Welcome to our Breakthroughs in Cancer seminar. And today's speaker is Ben Ebert, who will speak about the origins of cancer and clonal hematopoiesis. Breakthroughs in Cancer is one of our most important cancer seminar series at Stanford. It's about 1.5 years old, 2 years old now, and we bring high-profile speakers who can teach us about the science of cancer and translation of oncology. And now it's my pleasure to introduce Dr. Ben Ebert, who is the president and CEO of Dana-Farber Cancer Institute. Ben went to college at Williams and then was a Rhodes Scholar and did his PhD at Oxford. He returned to the U.S. and went to Harvard Medical School, was an intern and a resident at Massachusetts General Hospital, a fellow at Dana-Farber, started his lab at Dana-Farber, and he's made several fundamental contributions to cancer science. I'll just cite two of those, and we'll hear more about this in his talk. But Ben discovered the mechanism of action of lenalidomide, a key therapeutic that's used to treat MDS and multiple myeloma. He found that the way that it acted was by degrading Ikaros proteins. Ikaros are master transcription factors in the lymphoid lineage. And this not only revealed the mechanism of action of thalidomide and lenalidomide, but it also opened up the field of protein destruction as a therapeutic option. And then he also discovered clonal hematopoiesis, which is common in aging blood and relates to mutations that occur in blood stem cells and leads to an increased risk of blood cancers and cardiovascular disease. And we'll hear more about that from him today in his talk. He's received numerous awards, including being elected to the National Academy of Medicine, the Damashek Prize from ASH, and the Sjöberg Prize from the Royal Swedish Academy of Sciences. And then last year, he became the president and CEO of Dana-Farber. So it's my pleasure to welcome Ben Ebert to Stanford, where he has a lot of friends and former trainees. So welcome, Ben.
Thanks. Thank you, Steve. And absolutely, so many friends and colleagues and collaborators at Stanford. So it's been such a pleasure. And one correction on the introduction is I didn't discover clonal hepatopoiesis. Sid did all of that. Ravi did that. And so a little intimidating standing in front of all of them having to give this talk. But great to be here. And also, I not only, so many collaborators here, but even more coming with that. We have one more, my fourth former trainee who's going to be on the Stanford faculty starting in the coming months. So I thought I'd talk about clonal hematopoiesis today, and again, work that Sid really began. These are my disclosures. But I think we're all familiar and comfortable now with the concept in cancer biology that cancer arises from sequential acquisition of mutations in a cell that is capable of self-renewing and becoming and acquiring those mutations and becoming cancer. And only in rare circumstances does an individual mutation have the oncogenic potential to create a full-blown malignancy. And in the vast majority of cases, those initiating mutations, if they do anything, will cause a clonal expansion of cells that is pre-malignant and not transformed yet. This is the work that Sid did literally like 6 months after he joined the lab when he took on this question of whether there are pre-malignant mutations in hematopoietic stem cells. And we were able to collaborate with investigators at the Broad Institute, David Altshuler and colleagues who were just starting to do exome sequencing at large scale at the time. Looking at 17,000 exomes seemed mind-blowing in its scale. And they didn't think about mutations that might occur at low variant allele fraction and only thought about the germline inherited alleles. And Sid was able to look for the low variant allele fraction mutations in genes that are known to drive myeloid malignancies and found them to be far more common than we ever would have expected at the time, that up to 10 to 20% of people by the age of 70 have a large clone in their blood comprising on average about 20% of all the circulating blood cells. So these are massive clones of cells bearing mutations in genes that we see in myeloid malignancies, including acute myeloid leukemia. But unlike acute myeloid leukemia or other myeloid malignancies that generally have multiple mutations that are in known recurrent drivers, these clonal expansions tended to have a single mutation and only rarely had multiple mutations. And some of those ones with multiple mutations really had likely an undiagnosed myeloid malignancy. And in some of these studies, those individuals go on to a diagnosis within a very short span of time. Another interesting way we've been thinking about this is that we know better than we have previously, I think, how many true hematopoietic stem cells we are. And this comes in large degree from work from Peter Campbell looking at individual hematopoietic stem cells and all of the somatic mutations they acquire, which end up kind of barcoding every individual clone, which allows you to estimate the true number of hematopoietic stem cells. And it comes out to around 100,000. And we also can estimate the number of mutations that are acquired in driver, or a number of mutations that are acquired in the exome. And it's on the order of one protein coding mutation per decade per hematopoietic stem cell. And so if you put that together, we are doing quite efficient genome-wide scans, genome-wide genetic screens of our hematopoietic system and all of our bone marrows through our lifespan, and we will mutate every one of our genes over and over again. And that is a pretty fundamental shift from the way I used to think about cancer development. I thought that if you got a p53 mutation in a hematopoietic stem cell, boy, that was incredibly bad luck, and that may lead to a horrible, myeloid malignancy. But every one of us is likely to get one of those mutation in p53 and DNMT3A and TET2 and ASXL1 and all the other ones. And then the question becomes not whether you get one of those mutations or whether it grows out. And that's borne out in these, in when we try to talk about the prevalence of clonal hematopoiesis. If you use exome sequencing, you find mutations that are present in a few percentage of the cells. But if you use very sensitive error-corrected methods, which we starting to use in routine clinical practice to detect minimal residual disease in myeloid malignancies, you can start to detect these mutations in everybody. We will all acquire DNMT3A mutant clones. We all acquire these clones during our lifetime, and it becomes a universal phenomenon, which raises the issue that you need to define the state a little bit more in terms of the size of the clone, or else you're talking about something that is truly universal. So if we're all acquiring those clones, what the big question then is, what determines whether that initial single cell with a mutation becomes a large clone of cells that comprises a large portion of hematopoiesis, and which ones maybe stochastically go away or don't become very large. The selective pressure that we know the most about is chemotherapy and radiation. And these are actually the cases that we see the most at Dana-Farber at Stanford Cancer Center, because these are the patients that we see. They got chemotherapy for a solid tumor, they got radiation, and the clones that bear a p53 or a p53 pathway mutation tend to gain a clonal dominance in the face of that. And I'll talk about that more in a little bit. There's major sex differences in these. A number of the mutations are almost twice as common in males than females. DNMT3A mutation, the most commonly mutated gene in clonal hematopoiesis, is more common in women. It's not a massive effect, but it's incredibly highly statistically significant. And really, we don't know the basis for almost any of that. We've done a little bit of work on how estrogen can stimulate DNMT3A mutant clones, but I think for the vast majority of these, we don't understand that very well. Germline predisposition is a major factor. We know a number of inherited bone marrow failure syndromes that predispose to leukemia, and each of those inherited alleles has stereotypical patterns of initial mutations following their inherited risk factor allele. Smoking seems to be, it's a small effect, but it's a real effect. In some initial data sets, ASXL1 was more common. I'm not sure if that will bear out always, but smoking does increase the risk, though it's a very small percentage compared to other malignancies. And then the last points I wanted to make in the introduction is that this is very much not a phenomenon that is limited to the blood system, that in probably all of our organs, we are developing somatic mutations in all genes, but in particular within genes that drive malignancy, leading to clonal expansions. This is one of the first papers that showed that using a sliver of skin under the eyelid called a blepharoplasty and taking hundreds of tiny little biopsies within that and sequencing them and finding these somatic mutations. And I think one of the great innovations of this paper and subsequent ones that Inigo Martin Carina did was to create this cartoon system of representing the circles based on the size of the mutations and then the circles within that when there are multiple mutations in a single cell. But I think it's very evocative to show what the skin might actually look like. It's almost a lawn of clonally expanded cells with drivers of known non-melanoma skin cancer mutations like P53 and FGFR3 notch mutations. So these aren't just passenger mutations, these are proper drivers. What's different in the hematopoietic system from the solid tumor is that solid tissue has spatial constraints, and those cells may expand but have some limitation, whereas the hematopoietic stem cells has the potential at least to continue to grow and comprise a huge percentage of the entire hematopoietic system. This is even more scary to look at, but this is from the esophagus. We know that the risk factors for esophageal cancer are smoking and alcohol exposure. But even in young individuals in their 20s who have not had a major smoking or alcohol history have an enormous number of mutations already in the esophagus, and they include P53 mutations, which is the marker of really bad esophageal cancer or bad Barrett's esophagus. And you can see in the bottom line that in older individuals, even without smoking or drinking, but particularly with smoking and drinking, have just, again, almost a completely packed esophagus full of somatically mutated clones. So this is a phenomenon of aging, that we are acquiring somatic mutations in all of our tissues. It has implications for thinking about early detection, because it's not unique to have a mutation in one of these genes. These little clonal expansions likely don't shed DNA into the circulation that would show up in a GRAIL test, and that may be why the GRAIL test could work. But it is an issue that initiating mutations are widespread and in all of our tissues. So I mentioned that the selective pressure we understand the most is chemotherapy, and I'll tell one little vignette about that. But this comes up all the time clinically as well as additional research more recently. So when I trained, we talked about chemotherapy and radiation leading to therapy-related leukemias entirely because those are mutagenic therapies, that you introduce mutations into stem cells, and those went on to eventually sometimes lead to therapy-related myelodysplastic syndrome or acute leukemia, which are very, very difficult diseases to treat. And they are predominantly P53 mutated, much more commonly than de novo leukemias. And what has changed over the years since then is that recognition that the P53 mutations often pre-existed the exposure to chemotherapy, and that the chemotherapy activates P53, causes cell cycle arrest or death, and the P53 mutant cells have a selective advantage under that selective pressure and become more common, and that larger P53 clone can just sit there for years or decades and then eventually have a chromothripsis event or become aneuploid and lead to horrible leukemias. When we've looked at this in many settings now, but this was the first time we did it, these are lymphoma patients undergoing autologous stem cell transplant, so they'd gotten extensive chemotherapy. And because of that extensive chemotherapy and that selective pressure, even individuals in their 30s had a significant prevalence of clonal hematopoiesis or CHIP, that in their 40s and 50s, 60s, that prevalence had gotten even higher. So with getting close to half of all patients having these big clones. The mutations that occur most commonly, P53 is enriched, and there's a gene called PPM1D, which we didn't really focus on too much before the chip field evolved. But that is a regulator of P53. It's a phosphatase that dephosphorylates P53 and activates it. And that is arguably the mutation that is most powerfully selected for in the genome of individuals who are exposed to chemotherapy or radiation. And that those mutations in the blood of somebody who's gotten chemotherapy have a big impact on outcome. Again, these are non-Hodgkin's lymphoma patients who are getting autologous stem cell transplants, and having CHIP increases the risk of a therapy-related leukemia. That makes sense, a therapy-related myeloid neoplasm or TMN. But it also worsens their overall survival quite dramatically. And those effects are pretty big, and people do a lot of thinking now about patients who have quite marginal benefit from chemotherapy, and if they have a P53 mutant clone, whether that makes sense to treat them. It's a very sensitive question, because if somebody has curative therapy from chemotherapy, it's very hard to deny receiving that. But as we understand the risks better, we can weigh the risks even better for patient care. And then this is from a separate study that Coleman Lindsley did in the lab, looking at therapy-related myelodysplastic syndrome. And just as an illustration, if you look at the mutations that are more common in therapy-related disease compared to de novo primary MDS, P53 and PPM1D are the mutations that are most common in therapy-related disease compared to de novo, illustrating that at the end of the pathway, when the malignancy develops, these are the mutations that are highly enriched. So now I wanted to note that while we started this these investigations looking at myeloid malignancies, we can now think about multiple different types of clonal hematopoiesis. One additional type are the mutations that can occur that lead to lymphoid malignancies. And while there are some point mutations that we can see in the blood that increase the risk of lymphoid and not myeloid malignancies, most of these are copy number abnormalities. And this field have referred to them as copy number abnormalities. We call them copy number abnormalities in other settings, but they're somatically acquired gains and losses of chunks of chromosomes. And we know about a number of these because we do cytogenetics in all of our leukemia patients. So 5q, 7q, 20q lesions are present in some, though very rare, in the general population. And the presence of those known copy number abnormalities that drive myeloid malignancies massively increases the risk of developing a myeloid malignancy, not surprisingly. But much more common are copy number abnormalities that are associated with CLL. And those you can see in the blood of healthy individuals and dramatically increase the risk of developing a lymphoid malignancy. There are some of these that will progress to a lymphoma as well, or other lymphoid malignancies, but many of those cells are in lymph nodes and don't circulate very well. CLL cells by nature circulate. And so most of the copy number abnormalities we see are associated with CLL and increase the risk of CLL. I didn't put this vignette in there, but you can see that many of these individuals will go on to get MBL, and that enables us to use these very large sample sets, very large data sets of individuals with copy number abnormalities as a proxy for MBL, even though we don't have flow cytometry showing that there's a clonal lesion. They do tend to be clonal. So as we've tried to parse out these different types of clonal hematopoiesis, we first tried to define CHIP as an entity with a variant allele fraction of at least 2%. That was a relatively functional definition because that was where we could see those clones with good reliability. With normal blood counts, we wanted to make sure that these are not individuals with a myeloid malignancy. And then CCUS, or clonal cytopenia of undetermined significance, are the same definition with a cytopenia. And that was adopted by the WHO and International Consensus Conference a few years ago. In addition, I mentioned there are copy number abnormalities. There's no reason to carve those out as a separate entity other than the fact that we detect these using different methodologies, but it's the same idea of a different type of molecular lesion. There are point mutations in lymphoid genes as well, as I noted. And then microclonal hematopoiesis, where there are tiny clones, again, is a universal phenomenon. So we all get that. And then there are also clonal expansions without any drivers that we know about. And some of that could be drivers we don't know about, like non-coding mutations, but they also could be pure epigenetic clones, where that stem cell achieved a stable epigenetic state that has higher fitness than other cells and grows out. But that is more difficult to detect absent deeper whole genome sequencing. So, as Steve mentioned in the beginning, one of the most surprising parts of this field has been the association of CHIP as well as MDS with diverse inflammatory disorders. And this came from Sid's observation that patients with CHIP had a higher overall mortality that couldn't be accounted for entirely just by a higher risk of myeloid malignancies, which, while commonly fatal, are relatively rare, and there must be other causes of mortality in these individuals. And over the last 10 years or so, many papers have identified associations between CHIP and various inflammatory disorders associated with aging. And in general, these stories have looked at huge biobanks with hundreds of thousands of individuals, found patients with CHIP, and looked at various outcomes and seen an increased risk of these various outcomes. And in many cases, been able to validate that in model organisms. Another incredible finding from Sid's lab here at Stanford is that individuals with CHIP are protected, have a lower likelihood of developing neurodegenerative disease, including Alzheimer's disease. So that is a fascinating complementary finding to these increased risk of inflammatory disorders. A parallel approach to identifying associations between CHIP and various outcomes is to do a pure epidemiology approach. And Lachelle Weeks, when she was in my lab, took this approach. And she argued that if CHIP leads to various non-malignant disorders and myeloid malignancies are preceded by CHIP, you could look at individuals who got a new diagnosis of a non-malignant disease in the 5 years leading up to the diagnosis of a myeloid malignancy. So they likely had CHIP at the time. Those cells may be inflammatory, and we could look at what diseases they get. And the reason for doing that is we can use very large data sets. We have SEER is the national registry for everybody with cancer in the United States, and people over 65. That registry can be paired with Medicare data, and you can get all the diagnoses that those individuals had. So you can do this in a very complementary way to the way we've done the associations of people with CHIP with subsequent incident diagnoses of non-malignant disorders. So she looked at patients who developed MDS and CMML. She didn't include AML because of the rapid progression of AML, and we didn't know if CHIP would be present in the 5 years prior, and could compare to a large set of individuals without the subsequent development of MDS or CMML, and saw an increased risk of a variety of malignancies, including or non-malignant disorders, including cardiovascular disease, pulmonary disease, various metabolic disease, bone and joint disease, and just as Sid had found, a decreased risk of neurodegenerative disease. And if you look at those individual diagnoses, it actually lines up almost perfectly with the prior studies done in UK Biobank and other huge cohorts of individuals with CHIP who got incident diagnoses of non-malignant disorders. Even the effect sizes line up extremely well with that, with a relatively large effect size for development of NASH or fatty liver disease. In fact, those studies with imaging show it's not just fatty liver disease, but the inflammatory component of that, which is non-alcoholic steatohepatitis, and osteoarthritis, osteoporosis, rheumatoid arthritis, and gout. And again, a decreased risk of Alzheimer's disease. So from both directions, really robust findings. So in all of those cases, it's impossible to conclude from those pure human genetic studies the causality. In one of those genes, TET2, which we find to be particularly associated with a number of inflammatory conditions, the murine model has been an extremely robust phenocopy of humans, that the clonal advantage is recapitulated, but also the inflammatory consequences of TET2 mutations are recapitulated. So the first on the left is a model of atherosclerosis. These are mice with LDL receptor knocked out and mice that are fed a high-fat diet. In these experiments, the mice are transplanted with bone marrow from either TET2 knockout mice or wild-type mice or heterozygous knockout mice. And we can look at the development of atherosclerosis, and you can see that the red spots, those are oil red O stained atherosclerotic lesions, are more abundant on the right in the TET2 knockouts than the wild type. And then other studies from the lab have used the same model, the same approach, transplanting wild type or TET2 mice, TET2 or wild type bone marrow into mice and exposing to other inflammatory stimulants, including in COPD exposure to tobacco smoke, and the TET2 knockout. The mice with TET2 knockout blood cells get more airspace disease. And in the setting of a Western diet or high-fat, high-cholesterol diet, the TET2 knockout mice get liver fibrosis more than the wild type. So in each case, just the presence of TET2 mutant cells or TET2 knockout cells only in the hematopoietic system, not in the other cells, is sufficient to accelerate these inflammatory phenotypes. But it's important to note that each of these models have a major inflammatory stimulus, either extremely high cholesterol levels, tobacco smoke, or diet. And that's probably true in humans as well, that these phenotypes don't occur in everybody. They don't occur as a uniform matter of having these mutations in the blood. There needs to be a stimulus, and that there's a hyperinflammatory response to these stimuli in mouse models and in humans. So what might be causing this inflammation in TET2 knockout cells? And this has been something we've wrestled with for a while. And it's a phenomenon that's difficult to study in vitro because these cells need to differentiate in vivo. They need to be exposed to their own environment. So we wanted to do in vivo screens. And in vivo screens and hematopoiesis have traditionally been really challenging. And so we've turned to doing in vivo barcoded screens. And so in this system, we have guide RNA libraries, either genome-wide or smaller libraries, but with unique molecular barcodes so that every single virion, every single virus will have a unique barcode, and so every single cell that's transduced will have a unique barcode that we can track. And the reason that we do this is all of the previous screens we've attempted in hematopoiesis are confounded by the extreme heterogeneity of hematopoietic stem and progenitor cells. So you can see these are examples of two non-coding guides with no biological consequence. In the top, if a guide RNA infects a cell that just doubles once and turns into two cells, you'd have count two guide RNAs in the readout. But another biologically inert guide RNA might infect a cell that differentiates and generates many progeny. And so the top one would look like in a standard CRISPR screen as a guide RNA that decreases the fitness of cells, or the bottom one that increases the fitness of cells when, in fact, they're biologically inert. If every single virus that infected a cell has a unique barcode, you can see that both the top and the bottom would be a single clone, and so you can correct for the different proliferative capacities of any individual cell. And so we've done this both for understanding the clonal dominance of TET2 or other mutations, but also to understand inflammation. And so in this screen, we took Cas9 expressing mice, transgenic mice, that either were wild type or knocked out for TET2, harvested LSK, hematopoietic stem and progenitor cells infected with a barcoded library. And this was actually a genome-wide screen. As our inflammatory stimulus, we use zymosin, which is a component of a fungal cell wall, and extremely inflammatory, and is injected into the peritoneum, and that creates an enormous infiltrate of inflammatory cells in the peritoneum. And we could see, and we harvested bone marrow at the end, as well as the peritoneal cells. And the guide RNAs that are enriched in the peritoneum are permissive for inflammation. The ones that are depleted in the peritoneum relative to the bone marrow would be required for inflammation in the peritoneum. And when we did this, the genes that we found, I'm not sure what we expected, but were hugely enriched for genes that are involved in lipid metabolism. And so it was a very metabolism heavy result, including major regulators like ACLY and OGT, which I'll talk about later. But either looking at the individual genes or as gene sets, lipid metabolism was hugely enriched. And this actually linked even back to the very first experiments that Sid did in vivo with LDL receptor knocked out mice and TET2 knockout hematopoietic cells, where he saw sheets of foamy macrophages in many organs, which are essentially macrophages that are lipid laden, have accumulated huge lipid droplets in them. So these are the genes. They're very involved in the development of triacyl glycerides and other neutral lipids. ACLY is a critical enzyme that converts citrate to acetyl-CoA and starts that lipid biogenesis. But if you knock out any of the other genes downstream of that, including HMG-CR, targeted statins, fatty acid synthase, or others, that also blocks TET2 mutant inflammation. And these graphs are individual mice that were transduced with guide RNAs and doing single guide validation of this screen. So very robustly knocking out any of these genes involved in lipid metabolism blocked the relative inflammation you get with TET2 knockout compared to wild type. And if these are important, you might have changes in lipids in the cell. So we did lipidomics, and you can see it's almost only on the right side of the volcano plots, because there were a lot more lipids in the TET2 knockout than in TET2 wild type cells, but they were primarily triacylglycerides, diacylglycerides, and cholesterol esters that are all neutral lipids that were higher in TET2 knockout cells than wild type cells. We saw that in both mouse and human macrophages and in neutrophils. And then you can stain with dyes that specifically stain these neutral lipids. And you can see on the right, there are these lipid droplets that you can see in the TET2 knockout cells. And you can see that in both mouse at the top and in human in the bottom. So both in mouse and human, TET2 mutant cells accumulate lipid droplets that are neutral lipids. These are known to be hyperinflammatory, that they induce activation of the inflammasome and expression of inflammatory cytokines. If you look by RNA-seq at all of the gene expression changes with a TET2 knockout, they are largely reversed with knockout of ACLY, one of the regulators. If I have a good pointer, but if this is the gene expression pattern of TET2 knockout with a control guide, the expression of the ACLY guide RNA is quite similar to the wild type setting. So that whole inflammatory phenotype is reset if you block the lipid droplet formation. So what could be tying TET2 knockout to these lipid droplets? Well, if you do IP mass spec for TET2 or fragments of TET2, The protein that is most pulled down by far is OGT, and that is an enzyme, O-glycnacyl transferase, which adds this sugar moiety, the glycnac, onto proteins. And OGT is actually a metabolic sensor, so it regulates nutrient uptake in response to many substrates, and that raised the question of dysregulation of OGT, dysregulating a metabolic sensor may be important for why you get a metabolic phenotype in these cells. So we did some carbon-13 tracing studies and found that one of the pathways or one of the proteins that is most aberrantly or most actively taking up C13 and integrating it is the UDP glycnac itself. So there's just a lot of glycnacylation happening in the TET2 mutant cell setting compared to wild type, and that whole pathway was heavily enriched in the TET2 knockout cells compared to wild type cells. If you do either IP mass spec or IP Western for that glycnac moiety, you see a lot more glycnacylated proteins in TET2 knockout cells compared to TET2 wild type cells. So there's kind of widespread, perhaps dysregulated glycnacylation of substrates in the absence of TET2, which is its key binding partner in the nucleus. And then if you try to find how these proteins interact, we did a base editing screen and found a location that had, when you knock it out, you got dissociation of TET2 and OGT, and that is right where AlphaFold had predicted an interaction. We don't have a structure of this, but could demonstrate by a TR-FRET assay that is the location where a TET2 peptide interacts with OGT. And if we mutate that TET2 peptide with alanine mutants, we can obliterate that interaction. So we think that there's a specific interaction between OGT and TET2. We see that by IP mass spec, IP Western. We can recapitulate that in TR-FRET and have a domain that's important. And that loss of TET2 kind of liberates OGT to have abnormal glycnacylation of many proteins in a way that it wouldn't have without TET2. And while I don't think we have all of the answers of that story nailed down, I think that is evidence that the abnormal metabolism seems to be important. We did another separate screen for TET2 clonal advantage, and in this screen, we found that TET2 clonal advantage relied on mitochondrial function, and including a gene called NCOA4, which is important for free iron generation for mitochondria and other proteins by degrading the complex that stores iron. But you can see in a seahorse assay that both the basal and the maximal respiratory capacity of wild type or of TET2 mutant cells is greater than wild type cells, and knocking out NCOA4 returns it to that baseline. And the mitochondria of TET2 knockout stem and progenitor cells is different from wild type. There's more mitochondria based on mitochondrial DNA analysis, and there's more cristae per millimeter of mitochondria in the TET2 knockout cells. So all indicating that they are more active. And we've kind of put that together into a common model that in one screen, we see this lipid droplet formation. ACLY is particularly important. And if we knock it out, we return cells to more wild type inflammatory state. But those lipids are also substrates for mitochondrial metabolism. And if you decrease the number of mitochondria, the function of mitochondria, you similarly decrease the activity of the hyperactivity of TET2 knockout cells. So that is sort of ongoing work that we have on the mechanism of action of or the biological basis for why TET2 knockout cells are hyperinflammatory. But the clinical challenge that we have faced is our institution, I think your institution and many around the country have CHIP clinics because people are seeing these mutations and a primary care doctor certainly doesn't know what to do with a TET2 mutation in the blood, and most hematologists don't want to deal with that either. So there's a clinic to deal with it. But the key for this or any other premalignant states is to determine which individuals are at higher risk, which patients just need reassurance, or not even patients, but which individuals need reassurance, and which ones might be candidates for therapeutic intervention that could decrease the risk of progression to frank malignancy. We've known over the years from our initial studies and really robust through all subsequent studies that there's a number of features that are really powerful predictors of progression. One is genotype. Some genotypes are very benign. Some genotypes are associated with more rapid progression. Clonal complexity, having multiple mutations, is always worse than having one. The size of the clone, these are all very intuitive. Huge clones have a higher risk of progression than tiny clones. And the other one that has been as robust as anything is the MCV, so the RDW, the red cell distribution width, the variation in size of red cells, which we get in every CBC, every blood count that we get in our patients, and has been long associated with age-associated pathology, overall mortality, but is very associated with outcome in the setting of CHIP. So we use the UK Biobank split into a derivation validation cohorts, each with 200 plus thousand individuals. And first thing, we looked at all the genes that are commonly mutated in CHIP and their hazard ratio for the development of myeloid malignancy. And you can see relative to each other, DNMT3A is very low risk. The most commonly mutated gene in CHIP is very low risk, which is great. TET2 and ASXL1 a little bit higher. Splicing factor mutations, which have been really a bear to study in the lab, but they are fast-growing clones, which is hard to fathom in many ways, that messing with the core spliceosome that is required for splicing so many genes in the human genome actually leads to a very powerful clonal advantage. Some of the mutations that are associated with AML, like RUNX1 and IDH2, are also very powerful, and JAK2 is extraordinarily likely to progress to frank malignancy. DNMT3A is so low risk that it always raises the question, is it actually protective? And it's not. Individuals with DNMT3A mutations, shown in green here, do have a higher risk of developing a malignancy than individuals without any mutations, but the absolute risk is incredibly low. And so those patients are very, very low risk, and compared to any other mutations, they're low risk. So Lachelle used determine which of these each of these features are most important using recursive partitioning, what the weighting should be, and then instead of making a super complicated kind of black box scoring system, made a very simple scoring system that is very intuitive. Whether there's a high-risk mutation present or absent, how many mutations, how big the clone is, how the red cell distribution width, greater or less than 15, how big the red cells are, greater than or equal to an MCV of 100, and whether or not there are cytopenias and age. And with that, you can really develop very impressive separation between a low, intermediate, and high-risk group. So the low-risk CHIP barely increases risk, about a threefold increased risk of a very rare event of getting leukemia. So those individuals are 90% of everybody with CHIP and can probably be reassured and not seen by a hematologist, or at least not regularly. Intermediate risk starts to raise the risk a little bit more, 35-fold increased risk. And the high-risk group, which is closer to 1% of patients, is about a 350-fold increased risk. And so over 10 years, the likelihood of getting leukemia is more than 50%. So those are the individuals who truly have a very high risk of progression. And not only do they have a high risk of getting myeloid malignancy, they also have increased overall mortality. This is in the UK Biobank, which is a relatively young and healthy cohort. So the 10-year overall survival is very good, 96%. And having a DNMT3A mutation or having low risk CHIP doesn't decrease that very much at all, but intermediate risk starts to decrease it quite significantly with an 84% 10-year survival, and the high risk is only a 50% 10-year survival. So a major decrease in overall survival. So even though we didn't train the model in any way on overall survival, it's very powerfully predictive of overall survival and validates that those high-risk patients should be followed. This has been validated in a number of settings, and similar scoring systems have been developed, but this is the opposite in some ways of the UK Biobank. This is in a cohort of very sick patients with very high risk factors for cardiovascular disease and much higher mortality rate. But despite that, the scoring system works very well. The cumulative incidence of mortality is much higher in high risk compared to intermediate risk, low risk, or no CHIP. And the cause of death, the predominant cause of death is, as we've seen in other cases, is cardiovascular and more in the high-risk CHIP group. We see other causes of death, including solid organ malignancies, respiratory disease like COPD, and then the relative increase is highest for hematologic neoplasm, as we always see. So both in individuals who are relatively healthy or individuals who have high risk factors, these risk groups separate who is most risk for overall mortality as well as myeloid malignancy. And then this is from a paper, this is just an editorial we wrote for a paper that looked at lung cancer and CHIP from this Crick Institute, where individuals who had a lung malignancy, particularly if they had a TET2 mutant CHIP clone that then went into the lung and could be detected inside the sequencing of the lung tumor itself. So they called that tumor infiltrating clonal hematopoiesis, which is a subset of all the cases. Not all of the cases of lung cancer with CHIP in the blood had a high degree of tumor infiltrating clonal hematopoiesis, but those that did had worse overall survival and perhaps more rapid progression. And we have a lot of hypotheses about why this might be. One of the other really wild clinical trial findings, I think, of recent years is a study of IL-1 beta inhibition by an antibody called canakinumab. And in that, a 10,000 person randomized controlled clinical trial, inhibition of IL-1 beta with canakinumab decreased the incidence of lung cancer very dramatically and decreased survival from lung cancer or improved survival from lung cancer very significantly, even though the trial was designed for treating cardiovascular disease. And so these TET2 mutant cells elaborate IL-1 beta, maybe promoting that new incident lung cancer nodule and contributing to its progression, but we don't really understand the mechanisms yet. But there is some interaction that can happen between mutant hematopoietic cells and mutant lung cells. So I'm going to pause there and just note that that somatic mutations in driver genes are common in all adult tissues that people have looked at, that CHIP in the hematopoietic system is extremely common, very age-associated, and associated with increased mortality as well as many inflammatory diseases, that those clones expand in response to various selective pressures, and the one we understand best is chemotherapy and radiation, and that many of our patients who've been treated in our institutions who are now cancer survivors have those types of clones. That TET2 inactivation exacerbates these inflammatory phenotypes, both in humans and in mice, and that seems to be associated with lipid accumulation in hematopoietic cells in these mouse models and in human cells. And risk of progression to malignancy in individuals with CHIP can be predicted based on the size of the clone, the number of mutations, the specific mutations, and that's a very robust scoring system that many groups have found very similar scoring system based on similar parameters. And just again to highlight the incredible pivotal role that Sid has played in this entire field, and Adam Sperling, who will soon be a faculty member at Stanford, as well as a number of other people who are faculty members around the country now. So I'll pause there and take any questions.
Thanks for an amazing talk. Questions for Ben?
Hi, amazing talk. I had a question about the unknown driver clones that you were mentioning in the introduction. And kind of two related questions. Firstly, do you really believe in the idea of kind of an epigenetic clone that has no driver? Mm-hmm. And relatedly, do you think that question is really provable? Because each of these clones has so many mutations on it. Do you think it's provable whether or not any of those mutations are functional?
Yeah, that's a really good question. So I think it's definitely possible. It's very difficult to prove whether an epigenetic state is solely responsible for clonal expansion. When the Peter Campbell's group and at the Sanger Center has looked at all the clonal expansions that happen in a bone marrow, all the tiny clonal expansions, as we age, we can develop a kind of oligoclonal hematopoiesis. So all of our hematopoiesis comes from a limited number of hematopoietic clones. And there are many, many drivers. They see hundreds of drivers that we haven't annotated as drivers of myeloid malignancy before. And they use a DNDS analysis, whether the mutations they see in coding sequence lead to a coding change versus a silent mutation. And that analysis would indicate that those hundreds of other mutations have some functional effect because the coding change is more powerful than a silent mutation. And so that would argue that there's actually huge numbers of mutations with very small selective pressures that are actually drivers of at least clonal expansion, maybe not malignancy. And so that makes it even more hard to prove that an epigenetic clone is solely responsible because every one of those clones has many passenger mutations or some somatic mutations that are change coding sequence, and many of those may be driving the clonal expansion. So I think it's possible, but very difficult to prove that a true epigenetic state could drive a clonal advantage. And there are likely many, many other mutations that we don't normally consider driver genes that are, at least on some level, drivers.
Ben, can I ask a question from online? It's from Robert Deep. How do you recommend monitoring asymptomatic patients with high-risk CHIP, especially as it relates to monitoring the clone via molecular testing or blood marrow biopsy?
I would say there's definitely not a good answer to that, and everybody's struggling with that. But people like Lachelle, who was in my lab and now leads the Dana-Farber CHIP clinic, are developing various white papers and guidelines that at least you can start to discuss. And it's not different from many other types of cancer where we start where we don't really know what we're doing and you start to make guidelines. But it is some tiered system of CBCs every 6 or 12 months and genetic testing if something changes or at some fixed interval. I would say that those CHIP clinics are in large measure research operations where the samples are being banked. And so we can address those questions better in the future rather than something we can do now. In our case, it's a joint clinic with cardiologists. So they're also doing maximal cardiovascular risk reduction by standard methods. And then in those high-risk settings, there are a number of clinical trials that are open around the country for those individuals. They include things like IDH inhibitors for people with an IDH inhibition. They likely decrease the size of the clone. But there are also things like decitabine, which works really well, and MDS, could you decrease the size of the clone? A big question there is what outcome matters, because if you decrease the size of the clone, which you could evaluate in rapid, in a short time frame, then you could have a positive trial. If the FDA didn't recognize that as an endpoint and you have to wait for overall survival or development of malignancy, then it's a 5 or 10 year clinical trial and we won't get an answer.
Studies are
hard to do.
Yes. Back there.
Hello. Sorry. I'm sure you must have seen the recent paper from Karpova et al. With frequent blood donors who develop these interesting clones that are sort of erythropoietin sensitive instead of inflammatory, cytokine sensitive. And that raises the question of whether some of this difference in susceptibility of women to CHIP mutations could have to do with alternate types of drivers in people who, have to expand their blood volume frequently, and maybe EPO-sensitive. Has anyone looked at multiparity with respect to CHIP risk and the potential existence of other types of drivers that we don't look at?
It's a great question. I've actually wondered about that for many years. Other than DNMT3A, CHIP is much more common in males. MDS and AML are also male biased. Whether EPO drives hematopoietic stem cell fitness in any way has always been a question. There's Sid and I were just talking about, there's some really large studies ongoing now in sickle cell disease, which is an extreme hemolytic stress, chronic hemolytic stress, but also an oxidant stress with a lot of free hemoglobin around. And there's always been a subtle increased risk of leukemia in sickle cell patients as well. So that will come out soon. So I don't know for sure on the EPO part. I think there's also been questions about mobilization itself, people who have, Good Samaritans who donate their stem cells for transplant, whether they're increasing their risk at all. And that is a stimulant for probably hematopoietic stem cell division as well, or people who are frequent platelet donors, 100 plus times, and are probably generating more thrombopoietin, which is obviously expressed on hematopoietic stem cells and could alter fitness. So those are really great questions, and I don't think we have the answers yet, but I do think that there will be some stress, and like you said, the increased blood volume, it could be an EPO thing. The challenge there is that EPO receptor really isn't expressed on stem cells, but there are probably other other cytokines that go up in response to that chronic blood loss. So it's a great question. I think those are the types of questions we'll try to answer in the coming years.
Inflammation playing such a central role, have people studied anti-inflammatory drugs, and vaccination even may have an anti-inflammatory effect?
Yeah, so some of those clinical trials that are ongoing are targeting various inflammatory pathways, so no readout yet, but that is a thought. I think we don't know whether, even though these mutations can lead to inflammation, that whether eliminating the inflammation would alter the fitness of those cells or even cause a regression.
Aspirin trials in colorectal cancer.
Yeah, the aspirin trials have not shown any effect, other than there was an increase in bleeding in patients with CHIP. But yes, that's. But other than that, aspirin hasn't had a big effect. It's hard to know whether that will actually decrease the size of the clone significantly. And if it was neutral in terms of the fitness, that's going to be super hard to figure out to watch patients for long enough to make sure it's not going up or down that much. But those are good questions. Actually, given these metabolic abnormalities, it also may be that if you just lower cholesterol levels maximally, that's good enough. That's the substrate that's needed for these clones to expand. And so, and some of the clinical trials where patients are maximally treated with statins and their cholesterol is as low as possible, the association between CHIP and cardiovascular disease is much, much weaker. And so, the cohorts where patients have a high risk of getting more cardiovascular disease and probably still have high cholesterol, the associations are much stronger. So I wonder if that's the easiest way to do it. There's some mouse data on a variety of anti-inflammatories, including metformin. So there's going to be clinical trials in metformin as well.
Another question online. It's for Ben and maybe for Sid, and it's what is the potential mechanism for the decreased risk for Alzheimer's in CHIP patients?
I wish I knew. I don't actually know the answer. One can only speculate. So it could be that. So one thing that we do know is that the mutant cells seem to be able to enter the brain and differentiate into the macrophages of the brain called microglia. So it could be that the mutations alter the function of the microglia in some way. Perhaps they become better at phagocytosis, clearing up the stuff. Perhaps they're more resilient to stress, so they don't die off. They're better at proliferating. We don't know the answer for sure, but those are the directions that we're looking into.
So at least they're at the mutant cells are at the scene of the crime.
Thank you, beautiful talk from their discovery of their chip and their mechanistic insight and the prediction of the risk classification. And my question is the aspect of the treatment of chip. And there, after the risk classification, there is some high risk group there. And there, if you think about the treating chip, what type of the treatment direction in the future you are thinking that which is available? For example, try to eliminate that clone or control the selective pressures or targeting the specific clone for a vaccine or immunotherapy or whatever.
It's a big question, a hard one. I think the first thing is do no harm. And, for a pre-malignant state with a relatively, great outcome, the tolerance of risk is very low. You really cannot tolerate an intervention that has a high degree of risk. Things like lowering your cholesterol is very safe. Treatment with metformin, where almost every metformin trial improves survival, seems very reasonable. And then anything that's more does have a little bit more potential for toxicity would probably be limited to those very high risk patients. And one would hope that if you decrease the size of those clones, you decrease the risk of progression just by numbers, that you have fewer cells available to acquire the subsequent mutations. But those trials have been started. They're not as easy as you might think to enroll and complete them, because there isn't systematic screening for CHIP. And so if you start with screening a lot of people, and only 10 or 20% of them have the mutations, if you're in a reasonably, in most cohorts, you have to do a lot of screening to find those individuals, and you have to decide whether they want to be involved in a clinical trial, and end up seeing a lot of patients to enroll every individual patient into a clinical trial. So they're not trivial studies to do. And if you need to find only the high-risk patients, you have to screen a ton of people to find them. So as more institutions around the country develop CHIP clinics, there are more high-risk CHIP patients identified, and then institutions have to work together. So we've been working with through a grant mechanism called Breakthrough Cancer with MD Anderson and Memorial Sloan Kettering that have large patient populations, and putting that together. We're starting to put a bunch of patients on, but they're still, low single digits, low double digits patients. It's not like we're enrolling hundreds of patients in these things. So, but it's a good question.
One final question. Do we have one more here or no? If not, then thank you, Ben, for a wonderful session.